All this is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. Today we have one more exciting news for vaccines. So I'm sure that you have heard about the Moderna's announcement for their vaccine to be 94.5% effective. So I wanted to go over the data that they have shown. Plus, I wanted to compare Moderna to Pfizer BioNTech vaccine as well, because these are both messenger RNA-based vaccines. I also wanted to make sure that we can look at the side effects that people experience with Moderna's vaccine. I'm not going to go into the detail of the mechanism of the vaccine because I've done a complete video about that separately. So if you are interested, I'll link that here so you can watch that. So let's start a discussion. Hopefully you're all doing great, and let us start. Okay, so this is Drbeen.com. And here is Moderna's press release. I believe I have the link in the description. In addition to that, this is the page, and I have the link in the description for this as well. This is the page for Moderna's trial enrollment. Although this enrollment was complete by October 22nd, it is not open for further enrollment, but still you can go in and check various parts of this enrollment process there. And finally, I saw that this article in NPR is really good for Moderna's and then for Pfizer's comparison as well. So with this, let's start. So I'm going to present the data that is here. I've highlighted majority of the things over here. So let's start talking about them on my illustrations. So first of all, they are saying that their vaccine is 94.5% effective. What does that mean? So forget about what is written here. For a second, focus here with me here. There were 30,000 participants in this trial. This is a phase 3 trial. And the division was 1-to-1. That means half of the participants received the vaccine 2 doses, and the other half received a placebo 2 doses. And now out of these 30,000 participants, this is interim data, so they are still working on the remaining data. So in this interim data, 95 people became COVID positive. Out of those 95, 5 were those who were vaccinated, and 90 were those who were not vaccinated. So the difference of these two infected groups is 94.5%, actually 94.7%. So this is what it means to be 94.5% effective. Now, let's very quickly look at the study itself. The vaccine that they are doing, this is a messenger RNA vaccine, very similar to Pfizer BioNTech vaccine. The name of the vaccine is mRNA 1273. This analysis that we are going to discuss today is an interim analysis. That means the whole data is being analyzed and a final report has still to be presented. This is a preliminary investigation just to give you some idea for where do they stand. The phase was phase 3. Of course, the next phase is that once this phase is approved, then the vaccine will be out in production. Effectivity 94.5%, and the p-value is really, really good, lesser than 0.0001. That is, the chance of finding the same conclusion just by random is going to be 1 in 10,000 occurrences. What does that mean? That if this was just by chance, then if we do the same study 10,000 times, then one time you would get this kind of a result. So of course, this is a very strong p-value, very significant. Next step, getting ready for the emergency use authorization. So they're saying that as soon as we complete our data, plus the requirement for 2 months of safety, that would then go into the EUA process, and hopefully they'll get the vaccine out. This is really important. Why do I say that? And I know that there are some folks who do not like vaccines or who are apprehensive with the vaccine for the first time. Still, you can see that it is very difficult to get out of this pandemic at this time. And then minks and the other things, that the mutations are still happening. In human race, we are all stuck at this time. Yes, there are people who say this does not exist, this is a hoax, this is a fraud, this is, there are, even this morning I was on a TV interview, and there were so many comments. I was looking at the comments on the side where people were saying that you guys are just making up things and this is all a fraud and it is a hoax and there is nothing like this and it has never been isolated and so on. So not considering them, the current situation is really bad for us. I have been, remember I talked about the cool bean whose mother passed away 2 days ago. He is so much in pain. I spoke with him last night as well. I spoke with him yesterday as well. He's devastated. And then his family, they are devastated. So even one person, when they are lost, the whole universe of that one person is collapsed. So it is not an easy thing, and it is good that the vaccine is out there. It is good that majority of us is going to get the vaccine, and that would help us get out of this situation. Okay, continuing on. This is important. I think this was really an important thing that I liked about Moderna's data that they revealed. So if I go here, if you read this paragraph, the phase 3 COVE study was designed in collaboration with FDA and NIH, and then they have given a breakdown of the participants. And for your viewing pleasure, I have drawn that over here. So first of all, it was a randomized, placebo-controlled, one-to-one control to a vaccination study. The dose was 100 microgram per dose. There were 2 doses. Now, enrollment has been completed since 27 October. They are working on the data. Total participants were 30,000. They were all 18 plus. And check this out. Moderna specifically made sure that those communities and those people that are at high risk are well represented in the participants, so we can see what is the advantage for them. So they had 7,000 people who were 65 plus. So they wanted to make sure that they can understand how the vaccine would help them. So that is a high risk group. Similarly, they had another 5,000 people who were lesser than 65, but had some chronic disease, for example, diabetes or hypertension or cardiac issues or obesity, which made them at a higher risk or made them part of a higher risk group. Thirdly, we all know that the communities that are non-white communities, they are at an they are also at an increased risk. For example, African American, we know that they are disproportionately affected. We know Hispanics and Latinos are disproportionately affected. Same is the case with other races as well. So they had 11,000 people. About 37% of their participants were these communities. So out of them, more than 3,000 from African American community, more than 6,000 from Hispanic and Latinx communities. So this is what I really like, that other than other participants, they had folks with old age, they had folks with chronic diseases, and they had folks from various races that were in a high risk group. So total high risk patient percentage was 42%. So almost half of the participants, lesser than half of the participants, were from the high risk groups. That I really like. Now, what were the endpoints? What were they observing? They were observing, the primary endpoint was that will a person who is vaccinated, will that person get COVID-19? That was one endpoint. That was one thing they were looking for. A secondary endpoint was to see that if somebody does get COVID, will they get a severe form of the disease? So I actually believe that a vaccine that prevents us, that protects us from getting into a severe form is a good enough vaccine as well. If we get something like cold, common cold, and we come out of it, fine. I can take that bother over a chance of dying. So now if you check here, what was that they had 95 patients, so far the data that they have released for 95 patients of COVID-19. What was their breakdown? Look, for the primary endpoint, 90 patients or 90 cases were from the placebo group and 5 were from the vaccinated group. That is one. Second, I think you would like this. 11 patients who became severe, they were all from placebo group. There was no patient from vaccination group that became severe or severely ill. So somebody is saying that, is it costly? No. So I believe I had a TV interview this morning and I was talking about the prices. So I think if I remember it correctly, Moderna's vaccine is from $30 to $40 per dose. And Pfizer's vaccine, I believe, is $20-ish per dose. Johnson & Johnson is $10. And I believe that governments are subsidizing them. For example, I think in the US, we will get them for free. I think similar is the case for the other countries as well. So I think that this is a really interesting data. Although their data processing is not finished, they are still working on more data. There are 151 cases. They still have to look at the remaining cases and come back with the full data. But still, this is really interesting data. 90 from the placebo, 5 from vaccination, and the 11 who became severely ill were from the placebo group. That means that not only this vaccine reduces your chance of getting the disease, this also reduces the chance of having a severe disease. So that is good. Now, EUA request is next. So Moderna is saying that we are still processing the remaining data, and within a couple of weeks, they would finish that processing and then they'll go out for the EUA or emergency use authorization. And they have to finish the 151 cases, plus they have to provide 2 months follow-up data for safety. That is FDA's requirement. Now, this is also very interesting. I really thought this was interesting as well. And that is, out of the greater than 65 years of age group, 15 people contracted the disease. What I do not know is, out of these 15, how many were vaccinated versus placebo? Similarly, out of the high-risk communities, 20 people contracted the disease. Again, I do not know the breakdown, or they did not publish the breakdown of placebo versus vaccinated. 20 people who became ill, 4 were African Americans, 12 were Hispanics and Latinx, and then 1 was multi-racial, and 3 were Asian Americans. So this was a breakdown, which is also very interesting. Then, I think this is something that is interesting for all of us. This is why I spent some time drawing these doodles. I hope you like them. So here, the safety and adverse reactions. The first and most important thing is there was no significant safety concern. Even if there were some side effects, these side effects were subsided within a few days. So they called it, most of the side effects were short-lived. That's one. After the first dose, majority of the people, and by majority I mean all the side effects, 2.7% of the people, so imagine 15,000 people received the first dose, and then 2.7% had pain at the site of injection. That is it. After the second dose, and I'm going to ask you a question now, and I'll give you a chance to think about it while I explain it. Why more adverse effects after the second injection and not the first? So the second dose, after the second dose, there were 4.5% of the people experienced headache. 9.7 fatigue. And these numbers are not to be added up, because it is possible that the 9.7 that felt fatigue, out of them there were some people who also had headache. So these numbers, these percentages are not to be added up, they are just overall percentages for individual symptoms. Fatigue 9.7%, pain at the site of injection 4.1%, redness at the site of injection, which is called erythema, is 2%, and then myalgia 8.9%, muscle pains, and then arthralgia, joint pains, 5.2%. So these were the side effects or the adverse effects. Most of them, they call them grade 3, and most of them were short-lived. So now I'm going to ask you, I asked you a question. Why do you think after second dose there were more adverse effects instead of the first? So there is a question, there's a comment. It's a good comment that we do not know how long the immunity would last. And I would just answer in the same way that I've been talking about the immunity. SARS-CoV-1, 2 years immunity. In some people, even now, when they were infected then, we still see their T cells responding to SARS-CoV. So that is one. Secondly, now we have clarity that immunity lasts for at least 6 months. So I believe that the immunity, and I have been saying this since April, this is because this is how the science is. This is how our immune system works. There is going to be at least 2 to 3 years long immunity. So I believe Kurt is responding, delayed release of B cell maturation and fight at the site. Absolutely. So what is happening is very good, Kurt. What is happening is that the first dose primed our system and created immune responses that created some memory cells as well, some memory T cells and so on. So now our body is ready to fight. So when you give the second injection, a tiny bit of fight happens at that time as well, because body says, oh, well, there is an antigen in, I'm going to try to fight it. The good news is that the dosage is spaced in a way that the body has not really armed itself fully. And the first injection's dose is small enough that it does not make the immune system go crazy. So the second dose, booster dose, actually helps. But some symptoms may appear, which is actually a good thing. That means that body is responding. So there is a comment here, answer my question. So Asadullah says, can we use it after reinfection? You don't need to use it after reinfection. Why would you want to use it after reinfection? Or I think what you're saying is after infection. You don't need to. After infection, in a normal healthy person, you do not need to have the vaccine, because the virus itself is the vaccine. Virus itself is the antigen. So you don't need it. Yes, those folks who have immune issues or who did not respond correctly, there may be a need there. Kent says that there would be cross-reactivity producing autoantibodies to our own cells. No. The antigen that our cells are presenting is the spike protein antigen. It is not possible that we look at spike protein antigen and start killing our own cells or start reacting to our own cell. We can kill the infected cells, that is fine. We cannot just start creating autoantibodies by looking at an antigen. Very few antigens are such that they look like our tissue. For example, strep throat can cause rheumatic fever because the streptococcus has antigens that can look like our heart tissue. But these vaccines are designed in a way that these do not look like our tissues. So this is not an issue. Christy says that some folks did have repeat infections. Yeah, you're correct. And I've explained it many times that reinfection simply means the virus coming in us again. That is fine. It continues to drop into us all the time. Becoming re-diseased, meaning the disease appears again. Out of 40 million cases now, we have 100, 115. What was their real case? What was their situation? How did their immune system respond? What were their genetic factors? What were their health situation? So out of 40 million, 115, I would not worry about that. Yes. So Luffy, absolutely, France. Luffy just decided to agree with it. So Josh says that, would you take the vaccine and feel safe about it? Actually, Josh, when I looked at their vaccine, I wanted to go and get it. It says that it had been completed. I did not look at it before. So yes, I would take the vaccine. So this is a good question, Steve. Do you think that these mRNA vaccines will be more or less efficacious than the adenovirus vaccine? Eventually, both of them have similar mRNA. One is a lipid particle and one is the adenovirus. I would rather have a lipid particle that in itself does not do much harm and just the mRNA vaccine produces the antigen, but generally they should both be the same. There is a question from Jolik the Hero or Holik the Hero. What about the immunosuppressed people? How does the vaccine work for them? That is a problem, that immunosuppressed people may not be able to produce or mount enough response even by the vaccine because their immune system is not working. It depends upon the intensity of suppression. We did a talk a few days ago where a person who was immunosuppressed responded to virus and cleared it, but she took 74 days to clear it. And if somebody is absolutely immunosuppressed, then there is an issue. Okay, so going back here, so there's one more question here. In case of repeat infection, should the patient be vaccinated? It does not matter too much. So Shahida, they can be given a vaccine, but I would first want to know, why are they getting repeated infection? Is the test incorrect? Was the first test incorrect and then now they are actually infected? Or was the first test correct and this test is incorrect? The reinfection is very rare. So we have to first see why the reinfection, then you can decide to give the vaccine or not. So Edward says, it's a very good question. If your cells are made to produce spike proteins, what is the danger of these spike proteins remaining in your system? Look, the thing is this, when our cell starts making spike proteins, there are two things that would happen. Number one, our T cells, cytotoxic T cells and natural killer cells are going to kill those cells. Number two, the cells are going to present the antigen on their surface, which would then cause the immune system to start acting and start taking care of the vaccine itself, the antigen itself. So antigen will be cleared out, and the cells that have the antigen, they will be cleared out as well. No, so they will not continue to produce. So, so let me actually explain this. So let's say over here, we have a cell. This cell has the messenger RNA for the virus, and that allowed it to make the spike protein here. Then the cell presented the spike protein on its antigen presenting complex, MIC1 or MIC2 or both. And then the naive T cells come in here, or if this is MIC2, if it is MIC1, and I'm kind of assuming that you know this because I've been talking about it for a long time. If it is MIC1, then a cytotoxic T cell will come in here. Cytotoxic T cell would kill this cell. The naive T cell will not kill this cell. Instead, it would cause the immune system to become activated. And then the immune system itself, once that is activated, plus the macrophages are activated, they would clear out all the antigen and the antigen-infected cells. So antigen will be removed. The cells that have antigen will be removed as well, or the cells would heal. So back here, you're very welcome. Gamer1, Dr. Bean, if you had to take 2 doses of vaccine, what if you accidentally got the other dose from other company or another kind of vaccine? Is there a side effect? Not a side effect, but it is possible that both of those. It's an interesting question. If they both produce the same antigen, that is same spike protein, then it should work. But if they have different pieces of spike protein as their RNA, then there may be two suboptimal levels of antibodies against two antigens. So interesting question. Ashok says that can vaccine produce memory cell? That is the point. The vaccine will create the antigen to which the memory cells will be produced, and they would stay for 2 or 3 years. Over address says, what's the story behind DNA? That is just such a BS story. The vaccine is not going to go, and some people are asking, will there be nanoparticles? Can you imagine that we are trying to deliver nanoparticles in needles? No. And secondly, this vaccine is not going to go and alter our DNA. Changing our DNA is a very difficult thing. It is not an easy thing that you just make a vaccine and throw it in and it's going to go and start changing the DNA. That's not going to happen. Christy says that these adverse effects are really those we see with most vaccinations. What, if any, serious adverse reactions were seen in this trial? None so far. That is the important part, because the what is that vaccine? The one that was in Brazil as well. I think it is Imperial College's vaccine. They had a person die there, so they stopped it in Brazil. These vaccines, Moderna and Pfizer, so far, no adverse effect like that. No severe grade 4 and more adverse effect. So the efficacy percentage is misleading. Disappointing that you're not addressing this. Carol, this is how the efficacy is measured. Efficacy is not measured that you have, let's say, 15,000 people got the vaccine. Now you're not waiting for them all to become infected, because you can't. And so it's not that you want to say that 95% of the 15,000 have to become safe. What you do is, in both the groups, you count how many are the total cases that are created, and then see what percentage betterment is in the vaccinated group. And that is what they're saying. They're not hiding it. This is how it is counted. This is how efficacy for all vaccines are counted. I didn't understand this question. What is a carrier of mRNA to enter in the cells? Thanks. So these are lipid particles that are wrapping around the messenger RNA. So the lipid particle goes in the cell and takes the mRNA with it. So again, Nishal Gupta, this is just, imagine if I am the virus. The vaccine is only bringing the genetic code for my hand, for my spike protein. So when that vaccine appears in our cells, it's just a spike protein that is produced. The rest of the virus is not there. So there is no concept of carrier. This is not a virus that can replicate itself forever. This is not happening. It's not a complete virus. It's just a hand of the virus, the spike protein. That spike protein does not have the capability of continuing to replicate itself. So no, it would not become a carrier, the person. Khalil says, will it provide lasting protection? Yes, at least we know that 2 to 3 years. Guy Telfer, when was the experimental vaccine first administered? Months or weeks? I missed that if you previously mentioned it. I believe that Moderna started somewhere in March, April timeframe. Remember that we actually thought that they were doing pump and dump of their stocks by giving those news. So they had started earlier. I think they had some data collection in September as well for phase 2, and now they are in phase 3. Or they just concluded their phase 3, 22nd October. Chris says, any worries about adverse reaction in a patient whom had been previously exposed to the virus? I assume that people in these studies were pre-screened for antibodies. That will not be the case when mass vaccination starts. Same behavior as if the virus reinfects us. So imagine if I got infected by SARS-CoV-2, then I recovered. So now I have antibodies. If the SARS-CoV-2 will arrive in my body again, which it will, when I'll go around and I'll talk with people and I'll mingle with people, I am going to get the SARS-CoV-2 again. But now, because I have antibodies, I would very quickly take care of it. Same is going to happen with the vaccine as well. That let's say if I was infected and recovered, and now you gave me the vaccine, my cells would have the antibodies produced very quickly and clear out the vaccine. This is a very good point, Steve. Will the medical community develop a point system for people to decide who is at higher priority than other to get the vaccine first? That is, you have diabetes. I think that that is what's going to happen, because in the next slide, I want to show the differences in the doses available. So there would be some prioritization that may be needed. Okay, so James has a question. My concern about vaccine and a trial of say 2,500 is not proof of high efficacy. This is a trial of 30,000 because technically it will complete miss humans with vastly varying biochemical individuality in millions and millions of SNP, making humans unique. Its suggestion is it is safe, and we studied 4,000 people could lead to death, and had the test been in 5,000 people, safety is never guaranteed to all. James, so good news is that the current study was 30,000 people. The Pfizer study is also a lot of people. And then as they start rolling out the vaccine, this is called the phase 4 actually. They would continue to monitor and look for any adverse effects. But if the adverse effects with these doses have not occurred yet, when they have given it to all kind of people with comorbidities, with old age, with higher risk proportionality, then it is, you can start saying that it is safe. And that is the data that shows it. So one more question, then I want to show you a table and then we can stop question. So if our IgG is very high, there would likely be no reason to take a vaccine, correct? Yeah. So for example, if you don't want to take a vaccine and you had been infected before, or you feel that you had been infected, get your IgG. And if you have antibodies, then yeah, don't need to have the vaccine again. Okay, so one more slide and then we can stop, and that is this. Moderna versus Pfizer. Excuse me. Moderna versus Pfizer. So vaccine type, both are mRNA vaccines. That's one. So they are similar. Protocol, that when did they, this is interesting. Pay attention to this one. When did they say that somebody is positive? In case of Moderna, they said that if you have 2 symptoms of COVID-19 and you confirm you have a positive PCR test, then they would say you are infected. In case of Pfizer, just 1 symptom was enough to declare somebody positive. So that is a difference in the protocol. Days when they started counting the cases. So let's say they've given me the first dose, they would not count me in even if I become infected. 14 days after the second dose for Moderna, 7 days after the second dose for Pfizer. And this can actually tell you that Pfizer has slightly higher number of cases because number one, they said 1 symptom only and you're a case. And secondly, 7 days after the dose. And so the immune system may not still have enough time. So this was a little more stringent, but still not too big of a difference. Efficacy 94.5%, and this is 90%. And I believe this efficacy is because of these protocol differences. Storage, this is important because storage for Pfizer is a problem. The Moderna storage, 25 degrees Fahrenheit or minus 4 degrees Celsius. Look at Pfizer, minus 70 degrees Celsius. Ultra cool refrigerators are needed. Or minus 94 degree, below minus 94 degree Fahrenheit. So how would you do that? That means not only transporting it is a problem, storage is a problem, but keeping it at the end point of care is a problem as well. So that, I think, is a red flag for Pfizer. Potency. They say, the Moderna people say, that the drug, the vaccine, will stay potent for 30 days at normal refrigerated levels. I did not find this equivalent here, so left it empty. Warp speed. So did US give them money? US had a contract with Moderna to say we would buy enough doses. I think it was 100 million doses for $1.5 billion. Pfizer says we didn't have a contract. Trump says we had a contract. But I think this is true that Warp Speed did not give money to Pfizer as an aid. They gave money to them, say that, hey, if you make these things, we'll buy them. Dose. 20 million doses in 2020 for Moderna, 50 million doses by Pfizer in 2020. So total 70 million for US, 500 to 1 billion in 2021 for Moderna, and 1.5 billion in 2021 for Pfizer, but that is international. And remember that Pfizer has I believe they have a plant here in Michigan, where all the vaccine they'll make here is for US only. And then finally, about the aid, I believe Moderna received about $1 billion from a US aid company as well. I do not know about Pfizer. So that is the difference in some of the differences between the companies and their statuses. So this is what we have. I hope that you are safe and you are happy and healthy. Please like, subscribe, and share. If you would like to support my work, there is a link in the description to send donations, and I would see you tomorrow. I'm just going to look at some comments if there are any, and then we'll go. So Nina says that, ouch, MS shot right out of fridge hurts because it is cold. So Pfizer super cold shot must hurt like ouch. Yeah, you're right. You are very welcome. Very good. So guys, thank you very much. I'll see you tomorrow.